Welcome to the SWAYAM NPTEL course on food microbiology for safe and sustainable food systems. We were discussing microbial growth characteristics. In this lecture, we will be discussing nature of microbial growth in food, and we will be covering mixed population, sequence of growth, growth in succession or diuxic growth, symbiotic growth, synergistic growth, antagonistic growth, metabiotic growth, commensalism, predation. Now, mixed population. Normally, a food harbors a mixed population of microorganisms that can include different species and strains of bacteria, yeast and molds. Some species can be present in relatively higher numbers than others. The growth characteristics of a mixed population differ in several respects from that of a pure culture, that is a single strain of a species. Depending on the environment, which include both the food environment, that is intrinsic, and the environment in which the food is stored, that is extrinsic. Some of the species or strains can be in optimum or near optimum growth condition. Because of rapid rate of growth during storage, they will outnumber the others and become predominant. This can occur even if they are initially present in low numbers. This is often the case in foods kept for a long time under a specific condition, such as at refrigerated temperature. If the product is enumerated initially, it may show that the majority of microbial populations is able to grow at 35 degrees Celsius and only a few grow at 4 degrees Celsius, that is refrigerated temperature. If the product is enumerated after a few weeks of refrigerated storage, usually populations that grow at 4 degrees Celsius outnumber those that grow at 35 degrees Celsius, but not at 4 degrees Celsius. Another situation can arise if a food contains among the mixed population two species initially present in equal numbers and both growing optimally under the specific intrinsic and extrinsic environments of the food, but one having a shorter generation time than the other. After a storage period, the one with shorter generation time becomes predominant. Many foods most often get spoiled by bacteria than by yeasts and molds, because in general bacteria have shorter generation time. Thus, in a mixed population, the intrinsic and extrinsic environments dictate which one, two or a few in the initial mixed population will become predominant and produce specific changes in a food. These aspects are very important in the control of microbial spoilage of foods and in the production of bioprocessed or fermented foods. Sequence of growth. Among the different microbial types normally present in a food, different species or strains can become predominant in sequence during storage. Initially, depending on the environment, one or two types may grow optimally and create an environment in which they can no longer grow rapidly. However, Another type in the mixed population can find this changed environment favorable for growth and grow rather rapidly. This shift in predominance can occur several times during the storage of a food. The sequential microbial growth can be seen particularly in foods stored for a long time. If a food is packaged in a bag with a little air, for example, ground meat, the aerobes grow first and utilize the oxygen. The environment then becomes anaerobic, in which the anaerobes or facultative anaerobes grow favorably. In the natural fermentation of some foods, such as sauerkraut, 4 different bacterial species grow in succession, one creating the favorable growth condition for the next. The desirable characteristics of the final product are dependent on the growth of all 4 species in a specific sequence. To identify the existence of such a situation, it is necessary to enumerate and determine the microbial types at different stages of storage or fermentation. Now, the growth in succession or diuxic growth. Microorganisms that can metabolize 2 or more nutrients in a food, one preferred over the other and present in limited concentrations, show growth in stages separated by a short lag phase. Initially, a bacterial strain grows by utilizing the preferred nutrient, and after a short lag of adoption grows by utilizing the other nutrient. During each stage, the growth curve has exponential and stationary phases with the lag phase in between. An example is the growth of certain bacterial strains, such as some lactic acid bacteria and gram negative bacteria in fresh meat. A strain grows initially by utilizing the limiting concentrations of carbohydrate present, followed by utilization of non-protein nitrogenous, that is NPN, such as amino acids substrates. This figure shows that diuxic growth. In the figure, that this is there are 2 types of sugars present, the first lag phase, then the exponential phase. Again, and in this period the one preferred sugar will be utilized. After certain period, then lag phase will come and bacteria will again acclimatize with this condition, and then again exponential phase will occur. And this is the same thing with the specific example of glucose and lactose. When they are added, a lag phase is appeared and the growth by utilizing the glucose molecules, and after exhaustion of the glucose, again there will be a short lag phase, and after that the lactose will be utilized. Again lag phase will occur, again lag phase will appear, and this process goes. This is the example diuxic growth with a specific microorganism, that is the E. coli in medium containing glucose and lactose. So, this is the lag phase for utilizing the glucose, this is the exponential phase for utilizing glucose, again there will be a lag phase, and again a lactose will be utilized by that exponential phase of the lactose. Now, the symbiotic growth. In symbiotic growth, 2 or more microorganisms help one another in food. One type may produce metabolic products that the second type needs for proper growth, but cannot produce itself. In turn, the second type produces a nutrient that stimulates the first one to grow better. Symbiotic growth occurs between Lactobacillus aeruginosus and E. Escherichia faecalis in a minimum medium in which each bacterium is unable to grow individually. Since E. faecalis requires folic acid, which is produced by Lactobacillus aeruginosus, whereas the latter requires phenylalanine, which is produced by E. faecalis. Symbiotic growth can also occur in yogurt fermentation, where Streptococcus thermophilus produces formic acid from simple sugars, which stimulates growth of Lactobacillus burggericus and Lactobacillus bulgaricus and Lactobacillus burgerisus produces amino acids from hydrolysis of proteins. In turn, these products stimulate the growth of Streptococcus thermophilus. This figure representation of the interaction between Streptococcus thermophilus and Lactobacillus burggericus during yogurt fermentation and their effects on the product characteristics. These dotted lines that actually shows the EPS, that is the exopolysaccharides, it is hydrolyzed to facilitate the exchange of metabolites by establishing close proximities between the two species. What happens? The Streptococcus thermophilus that utilizes urea to produce ammonia, and this ammonia is required for the proper growth of the Lactobacillus burggericus. Likewise, this Streptococcus thermophilus that produces formic acid and low chain fatty acid, carbon dioxide and glutathione etc., which is required for the proper growth of and so on. And this EPS, that is the epoxy polysaccharides, that also required for gel formation by whey protein and the creatine proteins. And this lactic acid produced by Streptococcus thermophilus, that is required for the coagulation of the casein, that is the milk protein, and the peptides, amino acids, which is produced by both the organisms, that due to proteolysis of the proteins, that produces bitter product peptides, and both the organisms they produce acidity. The synergistic growth, this is observed during symbiotic growth of two or more microbial types in a food. In synergistic growth, each type is capable of growing independently and producing some metabolites at lower rates. However, when the types are allowed to grow in a mixed population, both the growth rate and the level of byproduct formation greatly increase. The increase is more than the additive of the amounts produced by growing the two separately. For example, both Streptococcus thermophilus and Lactobacillus delbrueckii subspecies bulgaricus, when growing in milk independently, produce 8 to 10 ppm acetaldehyde, the desirable flavor component of yogurt. However, when growing together in milk, 30 ppm or more of acetaldehyde is produced, which is much higher than the additive amounts produced independently by the two species and is necessary for a good quality yogurt. In the production of a desirable fermented food, two separate strains and species can be used to induce synergistic growth. So, how do these two bacteria synergize? It all comes down to nutrition. Lactobacillus delbrueckii is able to break down the proteins found in milk much more efficiently than the Streptococcus thermophilus, producing free amino acids and short peptides. Streptococcus thermophilus needs these to support its growth, converting them into new proteins and a range of other compounds needed for its growth. In return, Streptococcus thermophilus produces large amounts of folic acid and ornithine compounds that Lactobacillus delbrueckii cannot produce in large amounts, but which are critical for its ability to make DNA. Thus, by fermenting together, these two species are able to grow in milk far better than either could alone. This is the Lactobacillus delbrueckii. They all utilize the lactose, that Streptococcus thermophilus and this both utilize lactose to produce diacetyl lactic acid and acetaldehyde. Here also same products are there, but at the same time when they act on the casein, which is the milk casein, milk protein, they produce amino acids and short peptides, which is actually required by the Streptococcus thermophilus. Also, the Streptococcus thermophilus that produces ornithine, which is essentially required by this Lactobacillus delbrueckii. Also, folic acid, this is essentially required by the. So, by that way they grow, they help each other to grow. And this EPS, the both organisms they produce EPS, that is the extracellular polymeric substances, which actually help bacteria and other microorganisms adapt to their environment. Now, the antagonistic growth. Microorganisms can adversely affect each other in growth environment. Sometimes one can kill the other. This occasionally occurs among many microorganisms due to the production of one or more antimicrobial components by one or more microorganisms in the mixed population. Some gram-positive bacteria produce antimicrobial proteins, bacteriocins, that can kill many other microorganisms. As Lactococcus lactis subspecies lactis produces nisin that inhibits gram-negative bacteria and kills gram-positive bacteria. Antagonistic characteristics is important phenomena in the control of spoilage and pathogenic microorganisms in food. The production of lactic acid by lactic acid bacteria or sulfuric acid by Thiobacillus thiooxidans inhibits other non-acidophilic microorganisms. Similarly, the production of acetate and propionate by Salmonella enterica subspecies enterica, another subspecies enteritidis inhibits Clostridium lactate fermenters at low pH, that is 5.8. At these low pH values, considerable amount of acetate and propionate present as undissociated acids penetrate the cytoplasmic membrane and hence increase the cytoplasmic acidity. This inhibits bacterium. Now, this figure shows that how the environmental factors that actually helps in the antagonistic bacteria. These two antagonistic type of bacteria are there and they produce some metabolite products and antimicrobial peptides that actually that can be directly used in the plants or the animal foods. They can directly apply it as living cultures on livestock or the fresh produce this, or as the protective culture on ready to eat food products, like this is the ready to eat food products, or as a starter or proactive cultures in fermented food like this. They are expected to grow and produce antimicrobial substances in situ, displacing unwanted bacteria. Alternatively, food grade preparations containing antimicrobial product produced at industrial scale by antagonistic strains can be applied as biopreservatives or as food additives to inhibit transmission of foodborne and or spoilage bacteria through the food chain, that is all 1 to 4. Now, the metabiotic growth. Different types of microorganisms are present normally in foods, but the predominant types can cause own changes on foods during storage period. Initially, depending on the environmental conditions, 1 or 2 types may grow optimally and create an environment in which they can no longer grow. However, another type can find this change environment to be favorable for growth. The sequential dominance can occur several times during the storage period of a food. If a food such as ground meat is packaged in an airtight bag with a little bit of air, the aerobes will grow first and utilize the oxygen. The environment will become anaerobic, in which the anaerobes starts to grow. Most natural fermentations or decompositions of raw foods associate with metabiosis, such as in sauerkraut fermentation. 4 different bacterial species can grow in succession, one creating the favorable growth condition for the next one. The desirable characteristics of the final fermented product depend on the growth of 4 species in a sequence. Miscellaneous bacteria, chiefly Enterobacter, cloacae, E. coli, Erwinia herbicola and the coliforms grow first and produce acids that activate the growth of lactic acid bacteria. Second, Leuconostoc mesenteroids. Third, Lactobacillus plantarum, and the last acid tolerant Lactobacillus brevis grow. They increase acidity of product in order. Acidity increases with each group further until they are stopped. In final fermented product, high acidity and oxygen on the surface of product activate the growth of film yeast and molds and reduce the acidity. Raw milk spoilage at room temperature can also occur by metabiotic relationships of microorganisms. Raw milk at room temperature is fermented by coliforms. Raw milk at room temperature is fermented by coliforms first and produces acids. This acid activates the growth of Lactobacillus lactis. Next, the acid tolerant lactobacillus such as Lactobacillus brevis grows and increases the acidity further until they are stopped. Then film yeasts and molds grow over the surface of raw milk to reduce acidity. Finally, proteolytic bacteria can grow in raw milk such as Enterococcus faecalis subspecies liquefacens and Micrococcus caseolyticus. Now, commensalism. Microorganisms may not affect each other, but one microorganism use the substrate that is produced by others. For example, cellulose hydrolyzing microorganisms produce glucose and cellulose non-hydrolyzing microorganisms use glucose. The anaerobic thermophilic bacterium Clostridium thermosalum produces multi-enzyme complex to hydrolyze cellulosic substrates. Other bacteria producing cellulose hydrolyzing enzymes are Acetivibri cellulolyticus, Bacteroides cellulozolvens, Clostridium cellulovorans and Ruminococcus flavivescens. Another example to commensalism type of growth is the vinegar production. In vinegar production, yeasts such as Saccharomyces cerevisiae produce alcohol from sugar, and Acetobacter such as Acetobacter aceti and Gluconobacter such as Gluconobacter oxidans use ethanol to produce acetic acid. Vinegar involves the production of acetic acid. This figure shows that the yeast, that is the Saccharomyces cerevisiae, they act on the sugar compounds and they ferment that sugar compound, that is the like banana, the supply of surplus banana that contains the sugar, and when the Saccharomyces cerevisiae acted upon that, they produce the alcohol, and this alcohol is now served as the substrate for the action of acetic acid bacteria, and then they produce the acetic acid. Predation. The example of predation growth is the attachment of bacteria such as Myxococcus, Delobibrio, Diplobacter and Stenotrophomonas maltophila. Stenotrophomonas maltophila to gram-negative bacteria penetrating the cell wall and subsequently multiplying within the periplasmic space. Multiplication of predatory bacteria occurs with the utilization of cellular components of the host cell and leads to the formation of 4 to 20 daughter cells within 2 to 3 hours after infection. The host cell cytoplasm can be degraded by predatory microorganisms. These parasites differ fundamentally from viruses in that it does not depend directly on the metabolic machinery of the host cell. Delobibrio species and similar bacteria present in many different aquatic and terrestrial ecosystems.